All right, so Dr. Raquel Rodríguez-Pérez will be presenting the talk, Leveraging Molecular ML and Property Prediction in Drug Design. Dr. Raquel Rodríguez-Pérez is an Associate Director and the Senior Principal Data Scientist at Novartis Biomedical Research. In her current role, she integrates AI technologies into decision-making in drug discovery's project. She enables AI-driven drug design by leveraging techniques such as molecular property prediction, generative chemistry, uncertainty estimation, and explainable AI. Raquel obtained her bachelor and master degrees in biomedical engineering from the University of Barcelona and her PhD in computational life science at the University of Bonn. She was also a Marie Curie Fellow and worked at the computational chemistry data science team at Boehringer Ingelheim in Germany. Her research has focused on predictive modeling and pattern recognition for different applications in chemistry and life sciences. So I'm really looking towards that talk because that talk, I think, will open our eye into how to best use these kind of machine learning techniques that everyone is presenting, but in real world industrial scenarios. Thank you, Raquel.
Thank you. Thanks a lot to the organizers for the invitation, for the kind introduction as well. Looking forward to explain you a bit how we apply machine learning models in pharmaceutical research. In Novartis, we have been leveraging molecular machine learning for many years. I've been 4 years in Novartis, and I've seen a lot of applications in real-world drug discovery, and I've seen a lot of impact of machine learning, a lot of opportunities to accelerate and improve drug design. Today, I would like to give you an overview, but also highlighting the opportunities and challenges. I will highlight some open questions and I think relevant research areas from academia also that we can use later on in industry to improve our pipelines. So first of all, let me give you an introduction on machine learning in drug design, how we are trying to incorporate machine learning in our drug discovery pipelines. So first of all, I would like to highlight that the decision-making in drug discovery requires the combination of data, which can come from in silico, in vitro, or in vivo experiments. And you will have different amounts of data with different types of uncertainty at different stages of drug discovery. So you need to take decisions based on this. And machine learning is one of the ways of obtaining some of this information. And when we have some specific targets and we are designing molecules for these targets, we will need to score them according to some properties that we need to optimize. These designs can come from a human or from AI, but they will need to be scored in the same way. And actually, this property optimization and the prediction models that allow us to have an idea of the properties of the compounds that we are designing will be the focus of this talk. Afterwards, you can also make predictions of the synthesability, and you can measure some compounds either to try to find your development candidate or to improve your models in the so-called active learning loops. I'm sure you are all familiar with molecular machine learning for property prediction, but just to be all in the same page, these are the type of models that I will be talking about today. These are models that relate the structure of the compound to the property of interest. So they are called quantitative if they are regression, or qualitative if they are classification, structure property relationships. And for that, we will first need to represent the molecule numerically. For that, you can use more traditional methods such as fingerprints, where you encode atom environments, and you can also encode the presence or the absence of given chemical substructures numerically. And then you use this as input for a machine learning model. There are other types of representations, two-dimensional, three-dimensional descriptors, and the most recent learned representation that can come from embeddings of a deep neural network that has been trained on a large scale. For instance, one of the models that we are using a lot right now, it's the Melody. I don't know if you will be familiar with it, but this comes from a federated learning consortium of 10 pharma companies, where we trained models on a large scale, and we are able to use these learned embeddings from the Melody consortium to build our property prediction models. And actually, this will be one of the models that I will present today. Regardless of the type of molecular representation, the machine learning algorithm will relate the chemical patterns and the molecular representation to the property of interest. And it's really important to validate that the predictions that you are making are correct, and it's not only identifying correlations that are not able to predict for new series or new compounds. And now that we know which type of models I will be talking about, I would like to give you an example. This example is metabolic clearance. This is one of the main absorption, distribution, metabolism, and excretion properties, so one of a key ADME assay, in this case for metabolism. So we have some in vitro systems to assess the hepatic elimination of the drug. This is quite important because if we only focus on activity and we don't consider these ADME aspects early on, then we will have like a lot of chances of later stage compound attrition. So this is one of the models that I developed during my first years at Novartis. And I think the most interesting part is that we have a multi-species model because we had assays with similar protocols that were targeting liver microsomes from different species. In the table, you can observe that we had a lot of data for rat, but we had much less data for species like dog and monkey. So when we were building single task models only based on this assay data, we're having worse performance. And here you can see the results in the different panels. You have the different species, and we are reporting the geometric mean fault error, which is the relationship between the observed and the predicted. And we have compared different algorithms. We typically benchmark different state-of-the-art methods, in this case, XGBoost, a graph neural network on a single task, but we also built the multitask graph neural network and the multitask graph neural network ensemble. And especially for the species with less data points, we observed a benefit of multitask learning. I would like to highlight that the largest improvement was based on this multitask learning. So sometimes it's how you use your data and not the algorithm itself, because the representation either was the XGBoost with Morgan fingerprints or a single task graph neural network was not having a lot of difference in performance. And it was the leveraging of the different multi-species data that led us to an improvement. But this is only one property. We have a lot of properties to optimize. And for each drug discovery project, we will have a different target property profile. There will be a lot of commonalities, but also a lot of things that are specific for a project. For instance, if we are working on a project for a neurological disorder, we might want the drug to penetrate into the brain. So then our target property profile will be different. Maybe we have observed some liabilities for liver toxicity, and we also want to include this in our target property profile. So we have to define this together with the team, collect the required data, and build the required models that allow us to make good estimations of the properties. And for that, some things will be project specific, but the goal is to bring the ADME and PK properties to the target property profile, and if we are able to do it across all the projects. So now I would like to focus on some challenges and recent investigations that we did. And one of the aspects is this concept of the project specific versus something that it's applicable across projects. And this led us to benchmark different training set compositions. If we have large amounts of historical data in pharmaceutical industry, shall we use all of our data for model building? Or shall we focus on the specific parts of the chemical space that we are exploring right now? Sometimes the protocols of the assays change over time. So maybe if we are merging all the data together, we are incorporating noise, biases, we are modeling structure property relationships of parts of the chemical space that we are not interested anymore in. And I think everybody had a lot of perceived advantages and disadvantages on this topic. This was quite a hot topic at Novartis because medicinal chemists often were requesting local models for their project or their series because they had the intuition that a global machine learning model cannot capture the subtle differences of the structured property relationships. On the other hand, if you ask to a data scientist or a machine learning researcher, probably they will tell you the more data, the better. So what shall we do here? So what we did was a systematic comparison And we started building local models for the different projects and compared to a global model for ADME property prediction. And actually the comparison was quite extensive because we did it across 112 projects, 10 ADME essays, and approximately 330,000 compounds at Novartis. And I think the results are quite surprising, because in the x-axis you have the 10 ADME essays, and in the y-axis you have the relative performance. And you can observe that most of the distributions of the relative performance are on the positive side with all the dots in blue. Each dot corresponds to a project, and this is indicating higher performance for the global models. So there was a clear winner. The global models, based on heterogeneous data from different parts of the chemical space, different therapeutic areas, targets, were clearly outperforming local models for specific projects. And what it was also quite interesting is that only in 7% of the cases, the local models represented an improvement of 20%. So now every time that someone requests a local model, I send the link to the paper, which has saved a lot of time, but it has also given us some time to explore whether we can do better. And actually for that, we started exploring transfer learning techniques. Because at the end, we are having a changing distribution of our data over time. We are moving towards other parts of the chemical space, and sometimes even new modalities. So I would like to show you some of the results that we got with these investigations. Perhaps you have heard about transfer learning, and in the field of cheminformatics, normally it is applied for new tasks. So basically you have a matrix where you have in your rows the compounds, in the different columns your normally activity essays for different targets, maybe for this different essays for the same target. And then you have a new essay of interest or a new target of interest. You don't have a lot of data, and then you start applying few-shot learning, transfer learning. And I think this has been quite explored in this field. But what has not been so much under investigation has been, what if we include new rows that are from another distribution? Can we adapt our models for the same tasks, but doing something differently, trying to adapt the domain of our model? And this is what we have done for ADME models. We have set our global model with our global data across all the projects and also the historical data that we have in the database. And then we also took some project-specific data, which sometimes does not even have any data in the training set. Sometimes it has solely a small fraction, and then try to apply some domain adaptation techniques. And here I will show you some examples for one property and a couple of techniques, but in the publication you can see an extensive comparison with many more methods and essays. Here we come back to the metabolic clearance model that I presented earlier. If you remember, it was a graph neural network. It was coupled with a feed-forward neural network. So we have this graph neural network for representation learning, and then the feed-forward neural network for the metabolic clearance prediction. And the two transfer learning techniques that we tried, or the ones that I present here, are the weights initialization. So we consider the global model weights as initialization, and then with the project-specific data, we fine-tune the complete network, and the results are shown in orange. And the other strategy would be freezing the message-passing neural network, so the part of the graph neural network, and then we fine-tune the rest. Again, for comparison, we also report the performance of the local models, and this is relative performance compared to the global. So again, we observe that the local models are typically performing worse, but actually this transfer learning with the additional project data, it's allowing us to have lower errors. So this is also an interesting strategy in case a project is facing difficulties with our ADME prediction models, because we can further refine, and we also were able to see that there was no catastrophic forgetting in the performance of these models. And what about new modalities? We have focused a lot on small molecules, on the space of the rule of 5, but now we have a lot of new modalities that are also in our pipeline, and one of them are the targeted protein degraders. We have two main submodalities that are highlighted here, the glues and the heterobifunctionals. The heterobifunctionals have the linker and have larger molecular weight. So we were expecting that maybe they are outside the applicability domain of our models, and all the TPD programs at Novartis were not using our models because we thought they were not applicable. And actually, at some point, we thought, let's just evaluate, and if they are not good, maybe we can apply the transfer learning as well. And these are the results that I'm reporting here. In green, you can see the results, the mean absolute error for the different essays in the x-axis. And this will be across all modalities. In orange, you have it for bifunctionals, and in blue, you have it for glues. Interestingly, we observed that the performance for glues were consistently quite low, and in many essays, the models were performing very well on the glues. For the heterobifunctionals, we also observed quite a good performance for many of the properties. And if we look at the 0.3, which was actually mentioned in Cass talk earlier, and it's normally the number of the experimental variability. So normally it's twofold in general. So if we look at this threshold, we observe that only in some cases, model refinements might be needed. And in these cases, we also tested the transfer learning strategy with the fine-tuning. In this case, we tested two strategies. Both are fine-tuning on the complete graph neural network and feed-forward neural network. However, in one of the cases, we did it with the newest data. And in the second case, we did it with all heterobifunctional data. So we also exposed the model to some of the data points that were already in the training set, if they were from heterobifunctionals. And what you can see in the results, in the x-axis, you have the permeability, metabolic clearance for rat, for human, CYP inhibition, reversible, time-dependent inhibition, and reversible. So for all these properties, what we observed is that the performance on the global models, which is in green, was worse because this is the mean absolute errors. So the errors were higher consistently than any of the fine-tuning strategies. And depending on the property, one of the strategies of fine-tuning was better than the other, but there was not a real consistency across all the properties. So the take-home message from here is that we can adapt global models for specific projects, but we can also do it for new modalities. And actually, the QSPR models based on machine learning are applicable to TPDs, which is something that the community did not know so far. And another interesting aspect is that we are trying to work on ensuring that this kind of investigations can also be done in the public domain, and that there are high quality data sets. And one of the strategies that we started publishing are the surrogate data sets. So we cannot publish structures from our internal database due to intellectual property. However, what we could do is to use our high quality models that we have developed in-house to annotate the structures in the public domain. And we have done this already once. This is published. We did for a CYP inhibition model that we have internally, and it's published now in Chemical Research in Toxicology. We took more than 16,000 chemical structures from Campbell and PubChem, and we annotated them with CYP inhibition predictions from our in-house model. What is interesting is that when we developed a model based on this surrogate data set, the performance was quite comparable to our internal model. So this means that we can explore additional ways of model building in the public domain and have these open source tools for further research. And related to TPDs, we are also attempting at publishing a quite large data sets with more than 270,000 chemical structures from the public domain, including PROTACs. And we have annotated them with 25 properties. So this will allow us also to explore machine learning models for target protein degraders. And now I'm going to discuss the last challenge, which is one of my favorite ones, which is the uncertainty estimation. I think this is quite important because we often talk about uncertainty in terms of model predictions, but we also have to assess the uncertainty in our data. And something that we should do and we normally do at Novartis is to compare the performance of our models with the experimental variability of the underlying data, because this is the upper performance bound that you can get. But now let's focus first on the quantification of uncertainty in ML models. So normally the property models are evaluated on a test set, which can be challenging, which can be with a temporal evaluation. You will get some performance metrics, but then you will get a new data point. For this new data point, can you trust the model? And there has been a lot of research, and actually the concept of applicability domain is quite old in cheminformatics. A lot of people use the distance to the training set. This is quite a good proxy. Normally, if you have the chemical space, your training set is covering part of this chemical space and you have a new compound. If it's close to your training set, probably the model does well. If it's far away, probably the model has more error. But what is the problem? That sometimes there are discontinuities in the space. You might have a compound very close to the training set, but maybe it's an activity cliff, property cliff, you cannot predict correctly. And sometimes you also have compounds that are far from the training set, but actually the model can extrapolate. And these are the best cases for using the model. For instance, in generative chemistry workflows, if you can generate new molecules and these are different from your scaffold or different from your decorations and are predicted well, these are the most interesting cases. We want to capture them. So actually, these database uncertainty quantification metrics are not perfect, are giving us some information, but are just a proxy. Now we also have the model-based UQ metrics. You can have the variance from an ensemble, you can have the mean variance estimation, you can do some bootstrapping and get a variance as well. There are many options, but if you look at the performance of these uncertainty estimates, sometimes are also not giving you a lot of information about how the model is doing in real life. So for instance, for the coming back to the metabolic clearance model, we had an ensemble, if you remember. So we have also the ensemble variants available. And we used a calibration set. In the plot, you can see how the fault error was changing if we were adding compounds with increasing uncertainty or decreasing uncertainty. So the first one, you see how the error is going down if you start adding compounds that have a higher variance, and then you start adding compounds with lower and lower variance. So basically, yeah, it's encoding some information. Of course, if you focus only on the high variance compounds, you will have a higher error. And if you focus only on the low variance compounds, you have a lower error. We tested whether this was also the same for a validation set prospectively. And yes, we were able to have better estimations in the low uncertainty compounds. So if you check the table, you see R squares close to 0.6 for the low uncertainty, and for the high uncertainty, you have 0.4, 0.47. So yes, there is some signal, but this estimation is not perfect. And probably depending on the data set, the preferred uncertainty estimate will also change. Here, I also have a comparison to experimental variability. I think it's quite important to observe that when we have a low uncertainty estimation, we are actually quite close to the experimental variability, or at least the Q3 of the distribution of experimental variability. So again, when building the models, it's important to have in consideration the uncertainty in your data and the uncertainty in your model. But since the uncertainty estimation is quite a difficult topic, we have created a framework for uncertainty quantification benchmark. We are going to release this Python library that unifies the benchmarking of UQ strategies. So basically, when we went through the literature, there were many strategies for quantifying uncertainty, but also many strategies to evaluate whether the uncertainty quantification was good or not. And I think the evaluation will depend a lot on how you're going to use the model. Because if you want to use your uncertainty estimation to flag some compounds and put them out of distribution, then maybe you should focus on one metric to evaluate the uncertainty. But if you are going to do an active learning loop, and you want to create your acquisition function, you maybe need well-calibrated prediction intervals. So all these aspects are considered in the design of this library. You will have a comprehensive evaluation settings that are going to capture the superiority of uncertainty estimation metrics depending on your application scenario, and you will get some recommendations on which metric to use. So just to give you a quick overview, the only user input will be some information regarding the model, such as the predictions, or for instance, if you have any ensemble variance, or maybe the posterior of the Bayesian model. And you can also report some molecular representations. These molecular representations can be the ones from the model, or you can also decide to bring a CDD representation or molar. Like you can use whatever you want for representation. And then the library will calculate some uncertainty metrics. Some of them will be distance to a training set, kernel density estimations, and some of them will try to combine these two aspects of the database and model-based uncertainty quantification metrics. So we came up with some scores that are trying to bring these two aspects together, and we are currently benchmarking them using the library. Moreover, we have a special type of uncertainty quantification metrics that are the error models. So basically are machine learning models that predict the error of the property model. And these ones in our hands were also working quite well. But so far in our implementation, you need to take aside part of the data for the error model building, so you lose a bit of data for your property prediction model. And finally, as I mentioned, we have a quite complete evaluation module so that you have a nice benchmark of the uncertainty quantification metrics. And I would like to finalize the talk by moving towards in vivo PK predictions. Most of the models that I mentioned, or all of them, have been about in vitro ATME properties, but actually the in vitro ATME are also a proxy of in vivo PK in preclinical species that then will also allow us to predict better in vivo PK in human. The data that we have of in vivo PK in human is quite scarce and also very biased, because you can find some data in publication, but they are from commercial drugs and the chemical space that we are not currently exploring. So what we decided is to focus on predicting PK in preclinical species, because this data still covers the chemical space that we are currently investigating. And we have just put it on our Chem Archive a new methodology that we designed. This is to predict the concentration time profiles of the PK studies. So far, most of the investigations have focused on predicting clearance, predicting area under the curve, predicting derived parameters from the concentration time profiles. To the best of my knowledge, there was only one publication from AstraZeneca where they predicted a concentration at different time points, but these time points were specific. And what we came up with is a different strategy where we predict the constants of a PK compartmental model. So the compartmental model, it's quite used when you have the experimental data and then you want to fit it to do further simulations. Because once you have this compartmental model, you can simulate, for instance, multiple dosing. So in this compartmental model that you see in the slide, you have the central compartment, which is the blood, one first peripheral compartment that it's illustrating the highly perfused tissues, and then another one that it's illustrating the barely perfused tissues. And you have some constants that define ODE system. What we did is to predict the constants of this ODE system and then obtain the concentration time profile. The loss was calculated between this concentration time profile and the experimental one, and then we were back propagating. So at the end, you can obtain the concentration time profile, the compartmental model that allows you to do further simulations, and you can also derive the PK parameters. So far, we have evaluated this model for rat PK studies, and you see some examples of our predictions after IV administration and oral administration. Larger errors were observed when we were doing time split compared to random split, as expected, but actually the results were also successful for after IV administration in a temporal validation. Oral was more complicated, but you can observe in the table that the experimental replicates were also illustrating the high variability of these studies. Another evaluation was done after deriving from a non-compartmental analysis, deriving the PK parameters from the predicted curves and comparing the PK parameters to the ones obtained from the original experimental curves. So we have here results for area under the curve after oral and IV administration, maximum concentration after oral administration, and so on. You can get a lot more results and insights if you have a look at the publication. But what I wanted to highlight here is that these results were close to experimental variability, and also that the DeepCT method that I'm presenting showed similar or superior performance than when we were training the model to directly predict the PK parameters. And finally, as I mentioned, we can simulate multiple dosing because we have access to the compartmental model, and we also did some tests with commercial drugs to see how the simulations were looking like. So to finalize, I would like to give you some take-home messages. First of all, that machine learning models only based on structure are quite powerful tools for prioritizing compounds before synthesis, and also to drive the generation of new compounds, either informing medicinal chemists how their designs are looking like in terms of target property profile, or informing the AI system on what to generate next. We have presented a new methodology to predict the PK in vivo, specifically to predict the concentration time profiles. And these in vivo PK predictions are not only helping to prioritize compounds before synthesis, but also to prioritize animal studies. And I've also talked a lot about global models and transfer learning. The global models have shown generalizability across projects, even when the projects do not have a lot of data in the training set. If you apply transfer learning, you can get improved performance. And these models have also been applied to target protein degraders and have shown successful performance for ADME predictions. And finally, what is very important in drug design is to combine the experimental data and also the predictions, considering their uncertainty. You have different data available at different time points and different stages of the drug discovery process. And if you integrate properly the uncertainty and the variability that these have, you can have better decisions. And to facilitate unified evaluations of these uncertainty estimates, we will open source a Python library that is called Unique. I hope I gave you an overview on how we are using molecular ML methods in Novartis, highlight some of the opportunities and challenges, and thanks a lot to all my colleagues and all of you for your attention.
Thanks so much for the great talk. A lot of the work that was done here also resonates with some of the work that I've done during the past year. So I found it very interesting, and we kind of also arrived to the same conclusion that retraining on many tasks, even if like they don't necessarily correlate, even if they come from different protocols, it's really helpful for the downstream model. One thing that I found particularly interesting is the uncertainty estimation that you work with, and how you combine uncertainty, like distance of the data and uncertainty of the model. And I'm wondering, like, where do you see that uncertainty would be more helpful? Is it, at least at Novartis, is it like to do some active learning to reduce the uncertainty, or is it more to discard whatever the model is uncertain? Rather than like maximizing knowledge?
I think active learning is definitely quite important, but for most of the models I'm working with, we already have a lot of data. And even though active learning loops are useful, I think what it's more critical for me is to integrate them in decision making, to make sure that when decisions are taken with the models, it's because the model is quite sure about it. Also, when we are integrating models with different applicability domains in a multi-parameter optimization in generative chemistry, we need to start thinking more systematically on incorporating uncertainty, because if not, the algorithm is moving on directions that we are not expecting.
Okay, thank you. If there are any questions, please come to either one of the two microphones.
Great talk. I just had a question. So I thought the work that you had about the global property prediction, and also like training on different tasks, was helpful. The fact that that's helpful is interesting. I'm just wondering if you see like a lot of benefit from doing sort of more self-supervised or unsupervised training just on like pure sort of structured data, so data that doesn't require any assays.
Normally, we always do supervised learning, trying to learn from the essays that we have carried out in the past and trying to predict. On this sense, it has been more on representation learning, I think, what it has been studied by other teams at Novartis, but I have not been involved in that. When I use unsupervised learning, is more on exploratory data analysis, but not on predictive modeling.
Thank you.
Hi, great talk. You mentioned Project Melody and federated learning. I'm just wondering if you've developed ways to estimate data uncertainty when using federates.
Sorry, in case I didn't understand properly. So you are asking if we considered uncertainty when using the Melody embeddings or the Melody models?
Yes.
Okay. Okay. No, so far, actually, I have not used any uncertainty estimation from the Melody models. Now that we have established unique, we can run unique without having access to the model. So only with the predictions from Melody, we could run unique on it. And this is something that is actually in our plans, but we have not done so far because we're working on the library. And actually, I mentioned that I said during the talk that I will mention work with Melody, and the work that we did with the Melody embeddings was actually developing this. So I think it's quite interesting to highlight now that you asked, because the representation that we use for the chemical structure was actually the Melody embeddings. So from Melody embeddings, from a federated learning consortium of 10 pharma companies, then we were able to predict concentration time profiles of in vivo PK. So. But we have not propagated any uncertainty here.
Awesome. And unique, you're gonna open source.
Yeah.
Okay. Awesome. Thank you.
Thanks.
Hi. Thanks for your talk. I'm just wondering if for those global models, did you investigate different split methods, or do you just, because you have so much data, you can do random and it's gonna have the same impact, or do you need to cluster and ensure that you're picking up the most diverse set, for instance?
Sometimes for some benchmarking, we compare different splits. Like for instance, in the DeepCT here, you see that we also tested the random one. However, normally our gold standard is the temporal split. So we have the temporal information. We know when the compound was registered for the first time, and we also know when the compound was measured in each assay. So we normally do this time split to always ensure that we are evaluating the models in the same scenario in which they will be used.